thinking about it is that these early studies in man, need to really be designed from the perspective of answering again, taking words from nothing that David hasn't said, thinking about the scientific hypothesis and saying, is the test possible in the human, and can you do it? Yeah. Yeah.
Let me try that, because let me use Parkinson's as an example. There are a variety of alpha-synuclein therapies that are going to come forward. Now, obviously, the A beta may challenge that, okay? But as they move forward, the transgenic animal models with the synuclein overexpressors don't have very robust phenotypes, right? So they may or may not read out particularly well, but they're very hard to do and they're very late effects. At the end of the day, you may have a package that gets into humans, and let's say that we have, like we do now in Alzheimer's, an imaging agent for synuclein. If I'm sitting in a senior position in pharma at that point, and you bring me a package where it didn't work in the animal models, but at a safe dose, you were able to significantly reduce the alpha-synuclein, based on an imaging study in the human population, I'm going to invest in that asset. It's hypothesis testing. And so what I guess I'm saying is I'm moving the bar, the target validation bar, further down the food chain into actual humans. And in the past, we haven't been able to do that. We didn't have the tools. All we could do in phase 1 before was safety, and that's why so much importance was put in the animal models. As we now, and this is what a key point is, plan for this early, but if you can, in a human setting, using indirect measures, because not histopathology, demonstrate that things can move forward, there will be failures before there will be success.
I'm asking the same question. How do you make a go decision to do the millions of dollars before you go to humans if you don't have the answer?
I think the answer there is, there will be a pressing need to develop these new therapies. And one of the unfortunate things when we try to go out and look for new opportunities from pharma is there aren't that many very good ones out there. Okay? So if this were a field where there were hundreds of great hypotheses, hundreds of druggable targets, and then you're right, those that read out in an animal model are going to be prioritized, but we're not in the very broad sense of neurologic drug discovery. We don't have very many attractive looking candidates, and so strong hypotheses and good molecules are the ones that are going to get chosen to move forward.
So could I just add 2 points to that? I think one thing that might strengthen the package is really sort of thinking more carefully about the human genetics. Yes. And sort of helping that drive the selection of the target. And I know in some cases that's just really a minefield of its own and might pose challenges, but may not be. But it is something that I think you can hang a hat on and go towards the clinic for.
Okay. Well,
a pedagogical point that I just wanted to make is given the audience here, is that we often talk about target validation, and I sort of want to caution about using that word, validation, and when it is applicable to use it. So I actually sort of would campaign for or argue for that word. You don't really validate a target until you get into the human.
You get approved drugs.
An approved drug. And in fact, I would take it even one further step forward, and you could use the oncology example to say you almost need to get resistance to the medication and prove that the resistance is back in the target that you try to impinge in the first place to show that you validated the target. We don't have to put that bar in for neurological diseases per se, but still when we talk about target validation in the preclinical arena, I think it gets very squishy. So maybe it's more of a target prioritization exercise when you have multiple things that you're trying to assess in that arena. And okay, so the last point I wanted to make is coming from the government, and I'm at NINDS. I think the government also might have a role to play here in trying to move this bar. Specifically, I think if these kinds of projects need to move forward, there might need to be thought given to kinds of special funding opportunities to do that, and education of study sections to think beyond the bar of checking the animal model requirement for it to be funded. This is going to be, in a sense, just like your manifesto to sort of talk about doing rigor in animal models. I think you need to think about how we would fund these sorts of human experiments.
All right, one last quick question, because I see Ted is here. Please.
A kind of out-of-the-box idea, and I would like to comment on that. What about failure analysis of clinical trials?
Yes.
I mean, in other industries like
Yes.
aerodynamics, if a plane crashes, people go out and look for errors.
Two thoughts on that. One is, when we don't bring optimal molecules forward, and we go forward blindly, meaning that we don't really know if we achieve the right dose, the right exposure, to really know that a negative study was because we did not inhibit molecule X, is always a problem and always an issue. And that's why more focus on methodology in early development to know that at least the dose you've chosen for a definitive POC or later experiment is a true negative is critical, and that has not happened in the past. And the second thing is we need more data sharing. I'm very, very pleased to see the Alzheimer community starting to share data. You know, pharmaceutical companies realize that this is now a pre-competitive space and that they can share that data more widely. I think those two things together will improve our understanding of it and will move the field forward. But that, you know, that's still going to be a vexing problem when you're talking about assets that could be worth billions of dollars.
Like what about off-target effects of a drug? So the drug actually fails because it off-target effects who affect the functional outcome.
That's therapeutic indices again. You know, this is why the better your molecule is, you know, the better chance that you can reach the appropriate dose. And you know, and a good example of this are like the muscarinic agonists moving forward. You know, none of them had the requisite things to really test whether, you know, M1, M4 agonism was negative or not. You know, because we couldn't get to the right dose. So. Okay.
Thanks everyone for a good discussion here. That's exactly what cocktail hour is meant for. So I encourage you to continue it with Dave after he's had something to drink and can speak his mind freely. Dave, that was a great summary, and thanks for the lead into this next part of the talk, which is setting sail on the preclinical sea, taking us toward the shore of a clinical trial. This is the section on requirements for an IND. We spent a lot of time on the shore of research and development, and now we're going to spend a few moments pulling our way through a sea of dead souls and three-letter acronyms, which provoke a series of four-letter words. Can we start the timer here, by the way? I want to make sure we don't run over. Thanks. I need the angry red light to make sure that I don't run us into cocktail hour. So you've heard a number of these terms already. I just want to emphasize one or two before we get underway. The one term is GLP. It's been bandied about a bit. That stands for good laboratory practice. Or in some people's mind, it also stands for generate lots of paper. It is the transition from writing your data on a paper towel to putting this into a notebook that's signed, countersigned, materials that are lot checked, and moving toward a more documented style of development. The other three-letter acronym that I'll talk about more is GMP, good manufacturing process, or sometimes referred to as generate mountains of paper. But most of what I want to talk about today are these two acronyms, the CMC and the TOX, the two critical components in an IND package that the FDA is going to spend most scrutiny on. And so as we embark on this journey, it's useful to look to that patron saint of preclinical development, Dante, who wrote really the guide to preclinical development, The Divine Comedy. In the Inferno, of course, that dealt with the preclinical stage of drug development and probably early phase 1. When he got to Purgatorio, that really covered the middle stages of clinical development. And for those who are able to make it all the way through the 3 books and to Paradiso, that's the FDA approval. If you make it all the way through those 3 books, you'll see the important 15th century Italian term, SAFEM. This is the one word that you need to know in preclinical development. Now, for those of you who don't know 15th century Italian, I will translate this. Oops, if I get the right slide. Yeah. It's safe and sane. These are the 2 key components of the preclinical package that really the FDA is going to be focused on. Can you make this product consistently? Is it the same with each batch? And very importantly, is it safe to put into man? You'll notice that you don't see the word potent, efficacious. The FDA does want to see a rationale for what you do. But these are the 2 points that they're really going to focus on in that first phase. Later on, the efficacy comes in, though of course we all pray that we see at least a puff of smoke of efficacy in that first clinical trial. Now at this point, I've given you the entirety of the talk. This is a great time to check your emails. But for those of you who want to continue, we are here. And this is one of the few places in preclinical development where there's actually a defined stake in the ground. Because the rest of this process is a lie. There aren't discrete portions here. As you've already heard, they overlap. You really have to start thinking about this as you get started here. And each part of this feeds into the other. But when we look at this whole package and think about how we're going to get to this milestone, it's important to understand what is required for an IND. When do you need an IND? Well, most commonly you need it when you've got a new chemical entity, an NCE, that's not approved for the indication under investigation. Now, there are times when you need an IND for an approved drug. You need those for a drug administered at a new dosage level or drug administered with another drug in an unapproved combination. What I will mostly focus on today is this white bread, regular, first time in man, single drug approach. And with that, I'm also going to dispense with another IND. I just want to talk to you very briefly about what a phase 0 is. I'm mostly going to be talking about a phase 1, what it takes to get there. But the FDA, several years ago, developed this exploratory IND, which addresses the observation that we are not big mice without tails. That's really sad. I still think we might be big rats without tails. But what this approach does let you do is go into man with a minimum amount of preclinical data to test not for efficacy, but just some of the PK, PD aspects that we've already been discussing. Because other in vivo models and in vitro models tell you a lot about efflux, about crossing the blood-brain barrier. But if you have the right technique, you can still learn something by going into man and learning where your drug goes. As I said, there are minimal requirements, no real PK, single dose in rat, and safety is the main thing. And this dose must be non-pharmacological. So in other words, you must have some data that shows what you take to hit your target, and then don't approach that. This is not a safety trial, this is not an efficacy trial. Dose levels are typically very low, and what you're looking for, hopefully you've got a useful biomarker that can track where you're going, and you're looking at some indication that your drug is ending up where it should go. Now that preclinical program to get there can be very quick. You can get there in less than a year if you're nimble, and under $500,000 if you're lucky. But that is not a substitute for phase 1. So many people are not going to this because all this does is lengthen your timeline. But if you have reason to want to know whether something's getting into the blood-brain barrier or some other location, and you have a very sensitive tool for showing that, this can be part of the approach. That said, most of what I'm going to talk about here applies to the phase 1, getting into man and the true safety and CMC you have to do to get there. So, not quoting Dante in this case, but the other icon of drug development, Stephen Covey, and his classic piece, 7 Habits of Highly Effective People. What Covey says is you have to begin with the end in mind. And as Dave said in his previous talk, the end in mind is what your clinical label looks like, what your patient population looks like. So many of us in preclinical development like to form what's called the target product profile. It's something of a wish list, but it's something that reflects what our patient population is going to be, what our key characteristics we think are. Also, it's informed a little bit by some market analysis, understanding what the needs are, what the competition is out there. When the venture capitalists and others are talking about funding groups, they're starting to talk about not just proof of concept, but proof of relevance. They want to see that you have some idea of what you're targeting. Now, in something like Alzheimer's, that may be less of an issue. There aren't a lot of drugs in the armamentarium. But in MS, another drug that is anti-inflammatory is going to face a tough time. A drug that is neuroprotective or restores myelin would be a very interesting label, and you might want to think about how that would interact with the anti-inflammatory drugs. So that's your clinical end. Your preclinical end is really driven by what you need in the IND application, and there you see it not necessarily not to scale. It requires the following components. There are some introductory statements, there's some outlines, there's a general investigational plan, there are documents that show what your clinical protocol are. Study protocol, who the investigators running the trial are going to be, where it's going to be done, and importantly, an institutional review board that monitors the safety and the design. Any previous human experience is also in this, and additional information. The main things I want to focus on are sections 7 and 8 today. The chemistry, manufacturing, and control data, the CMC section, that talks about how you make it, that's the same section. And the pharmacological and toxicology data, that's the safe section. What you don't see here is an efficacy section or an MOA section. And again, not to hammer this too hard, it would not probably be a good idea to go into the FDA saying, we just had a dream, or it came to us as an inspiration, that it would be fun to try this in the brain. You want to have something in there to base this on. And the more MOA you know, the better you can inform things like safety measures and biomarkers. So efficacy is important, but for those of us academically trained, it's really hard sometimes to admit that this is really what they're going to be focusing on in the beginning. Now, ultimately, if a drug is safe but it does nothing, that's going to do no good. But the first thing the FDA is concerned about is that you do no harm. So let's talk for a moment about how you go from stuff to drug product. And the witch here has it right when she says, well, of course it's off. I have Newt is supposed to be off. She's following a recipe, and she knows what it's supposed to be like, and she does it reproducibly. So she's not just making a witch's brew, she's following the CMC. This translation here from bench to the clinic has several components as we go toward GMP, good manufacturing process. In the beginning, we've looked at a number of analogs. We've looked at a number of structural SAR relationships. We want more. Here, we want less. We want to narrow this down to ultimately one compound that goes in the clinic. In the beginning, we're not so much concerned about salts or polymorphs, and we think if we've got purity of 90%, we're really in good shape. The FDA doesn't agree. They want to see purity usually much higher. And if you've got impurities over 0.1%, you have to start showing that it's not toxic. And 0.05%, you have to report those impurities. So you have to not just characterize your drug, but all the other stuff that's tagging along for the ride. Stability is likely not well characterized in the beginning. You make it, you use it. Here, you really need to make sure that you have stability, at least for the duration of your clinical trial. And finally, although your grant may care how much this costs to make, you can have several convoluted steps and have a cost of goods that is unrealistic. But by the time you get to this stage of the GMP, you're really cost conscious. You really want to minimize the number of synthetic steps in a small molecule so that you can actually make enough of this and eventually market it. So we've talked a lot about small molecules today. I want to talk very briefly about how this looks to people developing biologics. Because there, what you have to worry about is proper protein folding, which is why so many of us work in mammalian cell lines, though there are cases now where you can use yeast. And in some cases, for simple peptides or proteins, you can also use bacteria. But what you're looking for is something that grows rapidly and at high density. You're really scaling up to tens of liters, hundreds of liters, not many, many, many Petri dishes. You preferably would like to see it grow in serum-free media, because that makes purification a lot easier when you don't have a lot of other protein gunk coming along, like fetal bovine serum. You want it genetically stable, you want to be able to do multiple passages, and you want to be able to recover cells at a high viability, so once you set your master cell line, you can come back to it over and over again, not start from scratch. And there are a couple of common lines that are used in the industry. CHO is still quite the workhorse. Some of these others also have high production, and all are amenable to batch-fed process, so you can keep putting in media, taking out your component, and then you have to purify it. Now, when you set this up, a couple things to bear in mind. If you're working with monoclonal antibodies, you're going to want to humanize those now, and that often runs into some licensing fees and other issues. Though purification can be quite easy, they're not always the best to get in across the blood-brain barrier, though Genentech now has shown ways to get antibodies across. That may change the game a bit. Certainly in cancer, monoclonal antibodies are one of the leading ways to develop new therapeutics. You want to start out with a well-characterized production cell line, hopefully one that you don't have to pay a lot of licensing fees for. Royalty stacking can make it really hard to get a partner. From this production cell line that has all the characteristics I just showed you, you're going to develop a master cell bank, freeze it away. You'll pull out aliquots of that and run your production, and then have a purification process at the end of that to get your pure molecule. In either biologics or in small molecules, you're going to want well-defined protocols. This is getting into the GLP and GMP process. Keep records of all the testing and validation of the key equipment. You're going to have to validate not just your materials, but how you make this. And again, start doing some stability testing so that you can show that not only can you make it, but it stays made for the duration of the trial. And then you're going to want to develop release specifications. How do you know when that material is ready for clinical trial? You look at aspects like identity, purity, chemical properties, and stability, and come up with a release sheet, a certificate of analysis. And you don't want to make that too tight so that you have to go through batch after batch before you've got something that you can use. But you want to make something that is at least standardized. And that requires validated assays. We'll probably hear more about this in the biomarkers and other studies to come. But validation takes on a different meaning when you go beyond research and into regulatory areas. because you have to do something you have to use an assay that's accurate, that is precise, that it has repeatability both from run to run and operator to operator, maybe even from lab to lab. It has to show specificity not just in buffer, but in the biological matrix that you're going to be measuring. You have to define what the detection limits are, show it has linearity and range, and show that this is robust. Understand what the limits are on variables like temperature. So good assays are the basis for getting a good material. And then the last part of this is the analytical testing. DP I used to work with, who's in development, used the phrase that I've always remembered as I look at this. He said, the FDA does not like mayonnaise. In the beginning, it puzzled me a bit. I thought, well, does that mean they like mustard? But what he meant was the FDA wants something that's characterized. So what you really need to do is look at all aspects of your compound from the identity standpoint, and this is where mass spec and NMR and other physicochemical characterizations come in. You want to look at its potency. You want a potency assay that shows you not just that you've got material, but it's active. You want a quantitative measure of potency. The quality to characterize that further, but most importantly, purity. And this is important particularly with your biologics that have a tendency to get contaminated by viruses. You need to have spiking assays to show that you have sterility, that you've got rid of pyrogens and microorganisms. And as I said, you want to do stability. Now, that's the CMC section. Fortunately, we've had some very good talks from John Seekars and others about toxicology. So I'm going to go light on that and move on to a few last mentions about project management and regulatory affairs. But what you want to bear in mind, as I said in the beginning, is a label that doesn't have a lot of side effects. And in fact, if you're dealing with cancer, you can tolerate some more of these side effects. I'm not so sure you can even tolerate pulmonary weevils or umbilical cord resurgence in cancer, but you can afford a narrower therapeutic index. For chronic indications in the brain, you want a very simple label with not a lot of side effects. So what you want to do, as was mentioned earlier in the session, is look at those those key target organs, CNS, pulmonary, and cardiovascular. There are some in vitro studies that you do early on, like HERG studies, to look at cardiovascular effects because they have been one of the reasons why a lot of drugs have failed. CNS, of course, is extra important in the neurodegenerative area. And as John said, you're looking for the NOAEL and maximum tolerated dose. A lot of this now at this stage goes into setting your clinical dose. What you want to do is think about frequency and potency of dosing. You want to start with a dose that's well under your MTD, and you want to build up slowly towards safety. Where you get worried early on is if you see a very narrow therapeutic index, or if you see these toxic effects are irreversible. Also, you'll want to be on the lookout during your tox studies for effects that are delayed or cumulative. The other thing that is worth looking backwards about is this whole issue of blood-brain barrier penetrance. Because just because you get into the brain doesn't mean it stays there. We also are looking more and more at in vitro models like MDR, MDCK, where there are efflux pumps that pump your material out of the brain. But lamotrigine and MS has also showed us we have to be wary of drugs that get in the brain and stay there. It's useful to bear in mind how long a drug stays in the brain, whether you get cumulative effects, and whether your tox program is going to take that into account. So as John said, you're generally doing this in rodent and non-rodent. Control groups, look at your vehicle, and generally 2 or 3 different ranges of doses. You're going to adhere to your dosing schedule and have a recovery period afterwards to see if there's a washout of any toxic effects. And you're going to analyze everything but the squeak. So as he said, there are a series of in vitro assays that also go into this. Most of them are looking for gross mutagenic effects. Those tend to be a bigger concern in cancer, and they tend to be a bigger concern with small molecules and with biologics. So with biologics, you generally don't have to do those, except possibly the micronucleus assay, until you're requested later on. But with biologics, you do have to take into account 2 other aspects. One is, if you're doing antibodies, the tendency for those antibodies to cross-react with other targets has certainly been a concern with some of the interferon approaches in MS that have generated neutralizing antibodies. So actually, that's more the immunogenicity testing. Here, you want to look for antibodies binding to off-target areas. That's been, again, less of a problem in neurology, but you run a panel of tissue sections to make sure that you're binding where you think you are. And again, in immunogenicity, you want to have an assay developed to start to look for neutralizing antibodies and how quickly they might come up. So in the last part of the talk, very quickly, as you build up your IND package, how do you prepare for these guys? And under the right circumstances, they're actually your friends. At least that's what they tell us. But they have seen it all, and they want to communicate that, and they want to have communication with you. And part of the trick is how and when you communicate with the FDA. The time to do it probably, if you haven't done this before, is not when you put together your package and ready to go into the IND. Some people talk about a pre-IND meeting. That can be hard to arrange. It's a lot more common to have a pre-IND meeting when you've gotten through perhaps some of your talks, want to check out your definitive talks, or certainly to check out your clinical protocol. So what you do there is you come up with a list of questions, submit them to the FDA, come with a plan. Don't come just saying, what should I do? They will look at the briefing document 4 weeks prior to a meeting, schedule it, and then have the discussion with you either over the phone or in person, and then follow up with a document that gives their recommendations. It's a good idea to listen to what they say. And in fact, as you prepare for that and beyond, the FDA does give you both the regulations, the CFRs, which spell out, as you saw in the table of contents, precisely what's required in each of those sections. And then they also have a series of guidances. They're suggestions, kind of the way the Ten Commandments are suggestions. You'd be well advised to take them under advisement. And they are looking at everything from the exploratory INDs that I mentioned about to content and formation of INDs for phase 1 drugs, updates on that, to some of the aspects of combining 2 drugs, to how to estimate maximum safe starting dose. So look to those recommendations to help you with guidance, because this is what you want to avoid. This is a clinical hold. This is the clinical equivalent of the black screen of death on your computer, except a lot more severe because it takes a while to overcome this. The sorts of things that trigger this are if the FDA thinks your clinical design is exposing patients to an unreasonable amount of risk, or if the investigators that you're proposing aren't qualified. Also, if you're misleading in your investigator brochure, the people that you line up to do this, and more it will require a larger and larger team of investigators to run these trials as they go forward. You need to make sure that you give them all your data, good and bad and indifferent, so they can assess the risk independently, not hide something, not mislead. And finally, insufficient information about the risks to patients. You can overcome this with the FDA. It takes a lot of communications and time. The best advice is don't do this. And that brings us to other disasters. The train wrecks that occurred with project management. When we talk about preclinical development, this often gets left out of the talk. And I think that as we talk about the reasons why drugs fail, sometimes we ought to give some thought to this reason. And that is, it takes a team. It takes a team of very different people to take a drug forward. And it often takes a champion and a project manager to make sure that that all comes together, that the trains don't just run to the washed out bridge on time. But if there's a problem in your development program, that you anticipate that and throw the switch and move forward in a different manner. There's not going to be a quiz on this, fortunately, and each of these flow diagrams is different. But what this does emphasize is as you think about the process of preclinical development, there are a lot of parallel activities that go on at the same time. You don't just do your CMC and then go to your talks and then develop your preclinical plan or your clinical plan. These all are integrated and come together in the IND submission. So what your good project managers will do is develop a Gantt chart with timelines, go, no-go decisions. And the industry average for doing this all from a standing start can be several years. And over $1 million, as much as $2 million, depending upon where you're starting. And all that's pretty daunting. So again, back to Dante. As Dante learned, when in hell, it hopes to have a Virgil. And there are many groups that can help you through this challenging transition period. We've heard a little bit today, and we'll hear even more tomorrow about NIH programs and about some of the foundation programs, such as the one I work with in the National Multiple Sclerosis Society, the ones that ADDF and Michael J. Fox run as well. They often fund this preclinical space and can bring in expertise as well. That's particularly important in the last couple of years where venture capitalists have migrated away from this early risky phase of development. They've figured out that the odds are not in their favor for their return on investment horizon. So they're leaving it to some of the public institutions to de-risk this a bit. And I think some of the programs we're starting to see with public-private interactions, like the one we're running with Serono, are also interesting ways for pharma to start de-risking this in anticipation of licensing in. There are also, as you've heard already, some contract research organizations. In rare cases, they may do some of this work for a piece of equity, but for most part, this is fee for service. That has some advantages. There are times when you want to collaborate with another investigator. That's great, but be careful what sort of IP entanglements that might cause. Might make it very difficult to get a partner to take things forward. CROs come in all sorts of shapes and sizes. Some are private institutions, some are very large, some are overseas, some are very new. I look at the directories to help you find what you need, but also when you're looking at a CRO, look for ones that don't have the violations when they've been audited, that have experienced the program before. Talk to who will be the project leader, not just the guy who gives you the talk on the phone, but who's actually going to run it, because these folks are often very good and very overworked, and you're going to want to work with them in partnership. You want to know that the group is going to be around for a while, that it covers it protects your IP, that you're not going to have this popping up overseas, that they're flexible, because a lot of this is not just color within the numbers and by the books. A lot of this requires some problem solving, because accidents do, or changes do occur in the plan. You want to look at price, of course. You also want to look at the quality of the report and information you get back. You want to make sure that you don't undercut on price to the point where you have to do this twice and it costs twice as much. And how you find these people is just network. Talk to other people who have done this, either within the academic world or in small companies. Talk to program managers. Talk to the foundations. They'll help you find these folks. So finally, drug development resembles one of the most famous maps in the history of cartography. This is a map of Napoleon's march to Moscow and his retreat. It's one of the original maps of attrition. Drug development is a game of attrition. The best we can do is try to line up as early as we can the strongest candidates we can, keeping the end in mind, and with that, realizing that with CMC it takes a scalable, reproducible process. With safety toxicology, it takes multiple assessments in well-validated models that reflect what you're going to do in clinical development. And finally, it takes a team. It takes a champion. It takes a team. It takes coordination. With that, brief thank you to some of my colleagues in the academic and industry world who have helped me on this. If I said anything right, it's due to them. If I'm wrong, I'll take the blame. It's a complicated process, and I hope this has helped you get at least the big picture. And I'll take a few minutes of questions if there are any. Thank you.
So I wanted to make a point about the phase 0 trials that you talked about briefly.
Mm-hmm.
And you made a point about the fact that people shy away from it because it tends to extend the timeline, because you end up having to do the other ones. And I think that's a fair point, but I maybe sort of offer a slightly different perspective, which is if for a particular individual investigator, that is in fact the case. But if you think about drug discovery in a space as a portfolio of activities, phase 0 might actually be a very helpful de-risking environment. So for the case of funding bodies, whether it be the government or foundations, it actually should be something encouraged to think about if they're funding a range of activities, that they encourage folks to think about phase 0 as a step, as a way of de-risking projects that don't spend millions subsequently.
I think it's a very valid point, and what I was trying to indicate was it's not a requisite, and what I would suggest for anybody who wants to use it is to make sure you have an assay that can make full use of that, but also bear in mind that when you have a drug process, the clock is ticking on your patents. And what you want to make sure is that you don't spend so much time up front getting the right answer, that you have a drug that is unpatentable because its patent life is limited. I've been in the place at a private research institution where we had a great cancer drug. It went through the RAID program. It had a great preclinical package. And the prospective partner said, we can estimate how long it will take for clinical development. And if we get through all of that, we'll only have 2 years to get our costs back. So you have to weigh in time and other factors. But for the right program, I think this is a very good approach. Because again, there are limitations to what we can learn in mice. They're useful. But for certain things like where does it go in the body, does it go to the brain, if you've got the right assays, this might be a very powerful technique. And that's why I didn't want to just do the standard phase 1 IND. So thanks for bringing that up.
The second point was about the target product profile. I think David referred to it as the evil thing, and I didn't expand on it when he said that. And my own experience from pharma is that I have seen many compounds die because of the target product profile. And so the point I want to make is that the target product profile, if defined but not revisited, it needs to be a living document that is constantly updated based on the changing environment, both science, market, etc. It's just from the viewpoint of those who are thinking about that. It's not something you say, oh, we did this and we're going to stick to it come hell or high water, because then you will be in Dante's Inferno.
No, exactly. You write that in clay, not in granite. So it has to evolve as you learn more about it. But I think it does make you think early. It's a useful exercise as long as you don't become wedded to it. It makes you think about some of the things you have to aim at. But you're right. If you kill, this is why I get a little worried about this kill early, kill often mantra we're hearing about. It's a tricky balance because you can kill too early. And if you put too rigid a target product profile, you give a target that you can't hit. On the other hand, I'm always hesitant to go the path of firing the arrow and then painting the bullseye around it. You really have to have something that you're aiming toward, and it has to have valid reasons for why you're aiming for it. So I think you're right, it is a balance. Good points.
So the last point I wanted to make was about what you ended with, which is to sort of emphasize this aspect of project management and its importance to the drug discovery journey. And in the many courses are taught in this arena, but seldom is there a section that you sort of spend talking about what project management really means, because that is probably, you don't even need postprandial glucose drop to have people fall asleep during that section. But it is so fundamentally important, and in fact, I don't know if it's worthwhile thinking about how there can be mechanisms to provide that kind of support, because it's not one that's commonly found in academic environments.
Exactly. Usually when I put that in, there's some project manager in the audience that jumps up and yells hallelujah because it's often forgotten. I sometimes include this in the talk. If you're familiar with Myers-Briggs analysis, it really made an impression on me in my first biotech company that people process information in different ways. And for instance, the person who's in charge of regulatory is going to look at a problem very differently than your basic scientist.